All right, it is time for the final step of cellular respiration. We're going to call it chemiosmosis. The oxidative phosphorylation is actually going to happen. And then at the end of this lecture, we're also going to talk about some other things that happen, for example, when oxygen isn't present. So stay tuned. All right, chemiosmosis. It sounds like osmosis of chemicals, and that's what it is. It is the movement of ions down a concentration gradient. Why don't we just call it chemical osmosis or chemical diffusion? I don't know, but this stage is called chemiosmosis. We're going to use this concentration gradient that we have built up with tons and tons of protons or hydrogen ions in the inner membrane space and very few in the matrix. We're going to use that stored energy, allow those hydrogen ions to pass through this really complicated protein complex, and that kinetic energy of their movement is going to provide enough energy to phosphorylate an ADP, add an inorganic phosphate, that's what the PI is, and create an ATP. This is what ATP synthase looks like. And you can see these little twisties. Those are the alpha helices of the tertiary structure of the protein. You can see some beta pleated sheets down here. The hydrogen ions are going to whoop down to this little tube here. This part here is going to do the actual phosphorylation as this turns. It's very complicated and super cool. I'll put a GIF in Blackboard. The hydrogen ions, they are diffusing down the concentration gradient. They're producing kinetic energy, and they're doing it fast enough that this can make 100 ATP per second in the best environment. And for every glucose molecule that gets used in glycolysis, we're going to produce about 34 ATP. It's actually a range depending on conditions, somewhere between like 28 and 38. So just, 30 something is all you would need to know. A lot of ATP. What happens though if oxygen is not present? If oxygen is not present, then this final complex 4 cannot bind the electrons to the oxygen and reduce oxygen and produce water, which means that it can't hand off its electrons. It doesn't have energy to push hydrogen ions through the membrane. The electrons don't get handed off, so the next complex can't hand off its electrons to cytochrome C, and since that one can't hand its electrons off, it can't take the electrons from ubiquinone, which means that ubiquinone can't pick up the electrons from complex 1. We get a traffic jam, and this whole thing shuts down. And we actually end up not being able to bring more pyruvate into the mitochondria. If this happens in the short term, we can do fermentation, which is our next step we'll talk about, and until we get more oxygen. If this happens in the long term as a result of, say, carbon monoxide poisoning, then the cells are actually going to start to die because they're going to run out of ATP. So carbon monoxide being a very unstable molecule, if you think about carbon wanting 4 bonds and oxygen only wanting 2, like what? There's a reason carbon dioxide is more common. The carbon monoxide is going to preferentially bind to this oxygen, hog all the oxygen, and not allow the electron transport chain to proceed. And that's how you die from carbon monoxide poisoning. Your cells run out of ATP because the electron transport chain stops. So be careful out there, please. Okay, if you're wondering how you get carbon monoxide poisoning, breathing exhaust from a car, or like using a grill or a hibachi indoors in the winter, they actually produce a lot of carbon monoxide in burning charcoal, so don't do that. Okay, anyway, oxygen, good. Without oxygen, glycolysis continues to happen. Remember, that's happening in the cytoplasm. What's happening in the mitochondria is no business of glycolysis. Glycolysis happens. We get NADH. We get a couple of ATPs. We can do a few cellular things with that. We get the pyruvic acid. If the pyruvates can't enter the mitochondria because the citric acid cycle and the electron transport chain are backed up, then it stays in the cytoplasm, and we have another set of reactions that use up the protons and the electrons in the NADH, and we create lactic acid. And you're thinking, wait, hang on, I know what lactic acid is. Lactic acid is that stuff that builds up in my muscles when I'm exercising. Yeah, it's the thing that causes the burn. Because it's a signal of your cells saying, we could really use some more oxygen in here. Now, it's not actually, your cells are not actually dying, even though sometimes they feel like they are. Your body is still breathing and still getting enough oxygen that your cells are not going to die. But some of the cells are doing fermentation, building up lactic acid. Your brain processes that as pain so that you will stop and breathe and they can get the oxygen they need. Now, animal cells can do fermentation for short bursts of energy. Some organisms do fermentation all the time, and that's the way they operate, and they can actually survive on just these 2 ATP per glucose molecules. Okay, so if you hear of a facultative anaerobe, that is something that, or sorry, not a facultative, an obligate anaerobe is something that always has to do fermentation. It lives within a low oxygen environment. All right, fermentation produces 2 ATP per glucose. We can do it for short bursts of energy. In animal tissues, including your body, it gets converted back into pyruvic acid by the liver and then gets sent back to cells to finish the citric acid cycle. Now, microorganisms, some of them can do that. A lot of bacteria and archaea live in hypoxic, low oxygen environments and survive completely fine on that. Now, yeast is a single-celled fungus that actually does a slightly different kind of fermentation, and instead of producing lactic acid, produces ethyl alcohol or ethanol and carbon dioxide from that process. This is a similar process, but ever so slightly different, and this is why yeast is key to human life, because without yeast, bread doesn't have bubbles. Yeah. We can't have beer. We can't have wine. Like lots of good things in life are because of yeast pooping out ethanol and carbon dioxide. Say thank you to the little yeasts. All right, so there are other ways to get ATP even when your tissues don't have enough glucose. So there are lots of amino acids that can actually go straight into pyruvate oxidation. Okay, there are some other carbohydrates and some amino acids and glycerol, the thing that is part of a triglyceride fat that can go, the glycerol can become a glucose and go straight into glycolysis. Some fatty acids and amino acids can go directly into the citric acid cycle. So there are these shortcuts, other ways that we can get molecules into the citric acid cycle, even if glucose is not present. Your body's going to really want the glucose, but if you're thinking, well, if I eat a low sugar diet, then my cells won't make glucose and then I won't store energy. No, your body has evolved to use any resource available. So this is an overly complex diagram showing the amino acids that can enter the citric acid cycle at different points of the cycle. And remember, every time we can enter here, we can then do oxidative phosphorylation and make ATP. And so that, my friends, is what you need to know about cellular respiration. So complicated. Oh my gosh. So please review that several times. And up next, we're going to learn about the opposite thing, photosynthesis. It's going to be cool.